Okay, welcome you all. Again, see to continue from the where I left from RT-PCR. See, once we have the screened RNAs, mRNAs in our hand, the next is to hybridize them to the genomes and find out which mRNA sticks to which nucleotide sequence. So that is done by using a more advanced technology called as the microarray technology. So there are a group of techniques which are called as microarray techniques, and a microarray technique or you can say microarray is a small silica gel or silica glass or plastic slide that is spotted with many different sequences of DNA. Is that clear? Many different sequence of DNA where each corresponds to a short sequence with a known, which has a known gene sequence. For example, if you take, there are many spots here, which one will correspond to the globin, another one will correspond to the actin, which is actually the cytoskeletal in nature. Okay, so, and this type of slides will have thousands of spots, can have thousands of spots in a small space. Okay, so how are they produced? So some are produced by spotting different samples of DNA onto a slide, much like the way we inject the printers with small dots with using a pen by using the PCR, or individually spotted on the slide. So the DNA fragments are maybe typically around 500 to 5000 nucleotides in length, and a few thousands or 10,000s are spotted. Few thousand or to 10,000s are spotted in a single microarray. So alternatively, microarray contains what? Short DNA segments, nucleotides that are directly synthesized on the surface of the slide. Okay, is it clear? Using a narrow light beams. So like that, the microarrays are prepared. So you can see in this diagram. Okay, where is that? So you can see a microarray consists of a DNA probe fixed to a solid support such as nylon membrane or a glass slide. Each spot has a different DNA probe. Each spot has a different DNA probe. So to this probe, what we do? We will collect RNA from the cell, convert them into cDNA using all the methodologies we studied till now, then hybridize them, see which mRNA is probing to which DNA target. Is it clear? So like this, we will be able to find the target easily using this nucleotide chip, which is called as the microarray. Okay, so, and I have given a detailed notes on this. You can go through that, and more importantly study the importance of this microarray, very frequently asked question. Is that clear? Okay, so next I'll continue with one more important aspect, that is, once we have identified the position, so that is the advantage we have with respect to microarray technology. In case of microarray technology, we can find the different, whether that particular mRNA is fitting to that chip or not, because already there are nucleotides to that these mRNAs will be fixed. So from that we will come to know the amount of mRNA present in that tissue or cell, to exactly note, yes, this has that or this may not have that. The next is to analyze the gene expression, which we use a methodology called as SAGE, serial analysis of gene expression. So rather than studying the complete cDNAs, there are some sequences which only expresses, and those can be studied much faster in nature. These are called as SAGEs, which is called as sequence analysis. This methodology is called as sequence analysis of gene expression. So then this methodology, short sequences as little as 12 base pairs in length, each of which represents mRNA, is present in a transcriptome. The basis of this technique is these 12 base pair sequence, despite their shortness, are sufficient to enable the gene that codes for mRNA to be identified. So a domain is more than enough. A domain of a mRNA is more than enough, because we can build more number of we can analyze more number of proteins in a limited space using a microarray. So that is the advantage we have with states. The should appear in the this is the calculation, with 12 base pair sequence should appear in the genome once in every 4 to the power of 12 times. Got it? So this is around 6. The average size of UKM is around 1500 base pairs. So 4 to the power of base pair is equivalent to the combined length of 11,000 transcripts. Got it? So this is a huge number of transcripts expected in all but the most complex transcriptomes, so that the 12 base pair sequence tags should be able to identify unambiguously the gene sequence coding for all the mRNAs that are present. So if we prepare small probes of this type, we can easily, and with high definition, we can easily find the target mRNA. So what we will do here, first the mRNA is immobilized in a chromatography column by annealing the poly A tails present in the 3' end of these molecules to oligo DT strands that have been attached to the cellulose beads, which I explained in the first video itself. The mRNA is converted into double-stranded cDNA and then treated with restriction enzyme that recognizes a 4 base pair target site and so cuts frequently in each cDNA. Very interesting and easy method to be applied. The terminal restriction fragment of each cDNA remains attached to the cellulose beads. So as I said, so you have to know that methodology well, which is very, very important, either in the magnetic bead methodology or cellulose bead methodology, by using gel filtration, enabling all the other fragments to be eluted and discarded. So when we build the oligo DT, instead of building the oligo T, what we do to that, we will bind a sequence of 4 to 12 nucleotides, which is having a specific 4 nucleotides base pairs like that of the restriction enzyme. Okay, so that 4 nucleotides will be repeated again and again, and they go and bind, and they can be easily identified. This is the advantage we have with this SAGE. So very important technique. Go into the detail of this technique and try to learn it. Is it clear? Okay, so these are the 2 important things we have to know. The next and last part of this is the massive parallel signature sequencing. Massive parallel signature sequencing. Remember, this is a procedure. Massive parallel signature sequencing. This is a procedure that is used to identify and quantify mRNA transcripts. Identify and quantify mRNA transcript, resulting in data similar to the sequence analysis of gene expression, as we learned in the last. That is, we use about 4 to 12 nucleotides with specific nucleotides like restriction enzymes, any restriction enzymes that can be, but it employs a series of biochemical and sequencing steps. So there it's direct. Either we connect with the magnetic beads or with gel filtration buffers, cellulose buffers. We attach 4 nucleotides which is specific to a particular mRNA and elute out. The rest of the mRNAs which will not bind it will be eluted out. Whereas in this case it's not like that. Steps massively parallel signature sequencing is sequenced developed in this methodology. This is the methodology which was developed by Berner, which is a bacteria-free based beaded bed-based library preparation, which uses the mega clone technology. So MPSS. So as you see, MPSS is a Berner which is who identified this, which is mega clone technology. You can see the diagram. Is a method which determines the expression levels of mRNA, by counting the number of individual mRNA molecules produced by each gene. So here we are finding the level of expression. and it's a open-ended technology. Is that clear? So this is the clone library. You can see a sample to describe. A sample of mRNA is first converted to a cDNA using cDNA, which makes which can be used for subsequent changes, manipulations. These cDNAs are fused to a small oligonucleotide tags, that is the sequence express tags, which allows the cDNA to be PCR amplified and then coupled with the micro beads, like we did with respect to SAGE. So this is a detailed procedure comparing to that of SAGE. Several rounds of sequence determination takes place using hybridization of the fluorescent labeled sequences, which will be around 16 to 20 base pairs from each bit. Fluorescent image captures the color and gives the signals, all of which will be effects to a two-dimensional surfaces. The DNA sequence are determined from all the beads in a parallel position, hence the name MPSS. There is, in some cases, approximately 1 lakh sequences reads are obtained per experiment. So it's massive, which can be used to identify more amount of mRNAs in less amount of time. So here the cDNA fragments are cloned onto a microbeads using the Linux mega clone technology, which is completely bioinformatics programming analysis, starting with 1 million molecules from a particular cell or tissue sample. Mega clone will produce 1 million bits, each containing around 100 to 1000 cloned copies of cDNA from each mRNA molecule. All molecules are covalently attached to the microbeads at their poly A ends so that they are available for sequencing reactions. Of course, this is a huge procedure. You have to go into the detail of that. So then thereafter we calculate the data, finding the abundance of the sequences and involve them in microarray assay. So what is the difference between the two, MPSS and microarrays? MPSS is notably the technology that has potential to capture virtually all genes present within the sample. This is very important to remember. Okay. Technology that has potential to capture virtually all the genes present within a sample and not just those that have been placed on the microarray. So this is the major difference, as I said, massive parallel sequencing tags. So, and this is like RAPD. We don't need any specification. We don't need any prior knowledge. We will just prepare the probes and we will mRNAs and we will hybridize them. Whereas microarrays has a limitation because we will be knowing very clearly which nucleotide we are positioning at which site or which position in the microarray. Got it? That is the difference. But with MPSS, the signature sequence is mostly untranslated, can be untranslated. As I said, there is no specification required. This is the major advantage we have with MPSS. So remember the advantage of MPSS very importantly. Using this methodology, as I given in the notes, 4,535 genes that are differentially expressed between normal brain and glioblastoma multi-tissue difference is identified. So you can see the difference and the opportunity we have with respect to MPSS. Even the tissue difference can be identified at once. Is it clear? This is all about the sequence determination by using the microarray techniques. So there are three techniques. One is the microarray, second one is the SAGE, the third one is the MPSS. So using all three means, using mRNA as the probes, we will hybridize them with the DNA and bring the genomic analysis. Is it clear? So remember very clearly, you may be asked with individual questions or a group with regard to all these three techniques.